we can group hormones that or the reproductive hormones into really four major categories. They can be neuropeptides, meaning that they are produced in the brain tissue. So in some part of the brain, they tend to be very short proteins or peptides. And examples of those two neuropeptides that are going to be key hormones in reproduction is GnRH and oxytocin. The second classification is a glycoprotein. And so these are proteins that are actually coated with sugar modules. And so the examples of those are luteinizing hormone, follicle stimulating hormone, prolactin, human and equine chorion gonadotropin. Sorry. The third classification is the steroid hormones. And the steroids are actually derivatives of the steroid cholesterol. And so testosterone, progesterone, estrogen, and dihydrotestosterone are all steroids that are actually derived or created from steroids. And so the last category is going to be prostaglandins. And prostaglandins are chemical compounds that are basically fatty acids. And so examples of those are going to be prostaglandin F2 alpha and prostaglandin E2 alpha. So as we look at the first chemical classification category, or the neuropeptides, I first just want to reiterate what you guys have already heard, that neuropeptides are really small hormones that are produced in the brain tissue. And so those, from a reproductive standpoint, are going to be gonadotropin releasing hormone that is produced from the hypothalamus, and oxytocin also produced from the hypothalamus. The mechanism, the way they elicit their actions, are different. GnRH is going to target the anterior pituitary, whereas oxytocin neurons are going to actually stretch down into the posterior pituitary and be released from that tissue. But regardless, because they are synthesized in neurons in the hypothalamus, they are small molecules. These hormones are small molecules with just a few amino acids. And so they're joined by peptide bonds. And so GnRH is the first sequence that you see on this slide. So this is the sequence of GnRH. It's really, really tiny. And this is also the sequence of oxytocin down here. And so these neuropeptides are produced in small quantities. They're a small structure, but they have a really, really big response. And they, as we studied last week with GnRH, or in part A with GnRH, it is really the regulatory hormone of the whole reproductive system. Our second biochemical classification group is the glycoproteins. And so as we look at the general structure of a glycoprotein in this slide, you can see that this is actually a protein hormone with carbohydrate moieties. And so carbohydrate is abbreviated as CHO. That's pretty common in literature. And so if you see CHO abbreviated in peer-reviewed publications, it more often than not is going to mean carbohydrate. And so these carbohydrates actually attach themselves to the alpha and the beta subunits. And with glycoproteins, these polypeptide hormones contain carbohydrate moieties, which cause the whole protein to have a very variable weight depending on which protein it is. So it could be several hundred kilodaltons clear up to 70,000. And so there's a large range here. And so some of the glycoprotein hormones are comprised of two side-by-side polypeptide chains. And so these are the one polypeptide chain, and this is the second one. And they're going to be bonded together to create the whole unit. And so those polypeptide chains have the carbohydrates attached to them. And the polypeptide chains are designated as either alpha or beta subunits. So in the anterior pituitary, that tissue will synthesize and secrete glycoprotein hormones that all have the same alpha subunit. So this alpha subunit is going to be the same for all anterior pituitary hormones. That means it's the same for LH, it's the same for FSH, and it's the same for TSH, which is thyroid stimulating hormone that we don't talk very much about in reproduction. And so the thing that makes LH different from FSH, even though they are both made and released from the same cell type in the gonadotrope cells, is the beta subunit. So that is going to be different for each unique hormone. So the beta and the alpha subunits are held together with hydrogen bonds. And so these are pretty weak hydrogen bonds that are held together with the van der Waals forces, if you remember that from chemistry. And so they are not covalently attached, which makes them easy to break apart and easy to stick back together. And so just keep in mind that the glycoproteins actually they're going to have to travel a long way. So this is examples of glycoproteins are LH and FSH. So they're going to have to travel clear from the anterior pituitary to the ovarian tissue or to the testicular tissue. And so the more carbohydrates they have attached to them, the longer their half-life will be. And so as we look at the function of these glycoproteins, what they are going to do to elicit whatever response they need to elicit is they are going to bind receptors on the cell membrane of their target tissue. And so in the case of FSH and LH in the female, as per our production scenario, those hormones are going to bind to that ovarian follicle tissue and to elicit its response. And so keep in mind that glycoproteins are not orally active, meaning that you really can't take a hormone orally, like FSH or LH orally, and they're also water soluble. So examples of glycoprotein hormones in reproduction are follicle stimulating hormone, which targets follicle stimulation or production, as well as sperm production in the male. Luteinizing hormone, which is going to trigger ovulation and will support the corpus luteum development after ovulation has occurred. Inhibin is a glycoprotein, and that is going to be produced from the follicle and will actually feed back to the brain tissue and suppress follicle stimulating hormone in the anterior pituitary. And sorry, I meant will feed back to the anterior pituitary, not the brain tissue. And then activin will actually release FSH from the anterior pituitary, and prolactin will be produced from the anterior pituitary as well, and it is going to be essential in maternal behavior in females. So as we look at how these glycoproteins function, this figure really just briefly shows something that we're going to study more in depth in the next couple of lecture videos. And so this is a secondary messenger system. And so when it comes to how these hormones function, we can take FSH as an example. So if FSH is produced and then secreted or released into the bloodstream from the anterior pituitary, it's going to bind its transport proteins, and then it will target its it's going to target its follicular cell on the ovary and is going to bind to the G protein coupled receptor here. And once that binding action happens, that is that cell surface receptor. And so that binding action is then going to kick off this whole series of events inside the cell. So I just wanted to point out why it's important to just remember that these hormones are going to bind to receptors on the cell membrane. And so FSH here is shown, or is represented by that red circle, and then the cell surface receptor is going to be in the middle of the lipid bilayer of the cell. It's going to be embedded in there, and that's going to be the receptor that receives the hormone that's going to kickstart the secondary messenger system.